Throat cultures. The microbiologist Lament. I used to think that kissing you was the nearest thing to bliss, until the doctor told me the dangers of a kiss. But now I know biology and sit and sigh and moan. 10 million bad bacteria, and I thought we were alone. The human mouth has the most numerous and varied bacteria of almost any location on the body. There are aerobes and anaerobes. Primarily, you have Viridae streptococci, that's the alpha strep. But there are all kinds of gram-positive cocci, gram-negative cocci, gram-positive bacilli, and gram-negative bacilli. And even a few spirochetes, yeasts, and protozoa roaming around in there. All of these are considered normal flora. They're commensals. They don't really hurt you. They live easily in your mouth. Some of them are even protective. The fact that they're growing there prevents other things from growing there. There are a few diseases that are fairly common in the mouth. One is thrush. Thrush is a secondary infection. Candida albicans is normally present in the mouth in small amounts. It's a yeast. However, after broad spectrum, high dose antibiotic therapy, you can wipe out pretty much all the bacteria in your mouth. Also, immunocompromised hosts may have altered amounts of bacteria in their mouth. If that's the case, the yeast can overgrow the bacteria and cause a secondary infection. A secondary infection is when something that normally should be there is growing to outrageous numbers, and that's what happens if you kill off the bacteria in the mouth. Typically, we don't culture for thrush because the symptoms are pretty consistent. There's a white coating on the tongue, and usually there's a history of recent antibiotic therapy, or it's a very young person, a very old person, someone who's had chemotherapy and is immunocompromised. There's that kind of history that goes with this. Dental caries is the most common disease worldwide. This is caused by your normal flora because your normal flora will produce a biofilm on your teeth. As the organisms in the biofilm strengthen, they begin to break down the sugars in your mouth and make acids which etch the enamel. As the enamel becomes damaged, then bacteria can invade the softer dentin of the tooth. Regular disruption of the biofilm by brushing your teeth and regular cleanings to remove the harder plaque helps prevent dental caries. There is only one true pathogen in the mouth, and that is Streptococcus pyogenes. This is not normal flora. This is not normally found in your mouth. It gets in and it makes you sick. You'll remember that Streptococcus pyogenes is beta hemolytic and classified as group A strep. This is the cause of strep throat. Certain very virulent strains of strep throat can cause rheumatic fever, and because strep can produce large numbers of antigens, you may get a type 3 hypersensitivity, that immune complex hypersensitivity that can damage the kidney, and you get glomerulonephritis. The classic symptoms are a red throat and pus pockets. You can confirm the diagnosis with either the rapid strep test and a culture. To do the rapid strep test, and there are several different kinds on the market, but they generally work the same way, you want to collect your specimen on a swab and mix the specimen in a reagent to remove the organisms from the swab. The reagent organism mixture is carried up a filter paper strip by capillary action. There is a band of antibody-coated beads on the strip. If the specimen contains strep A antigen, it will bind to the beads. The beads are continued to be carried up the strip. Further up the strip is a band of antibody. Those beads that are coated with antigen will stick to the antibody band, and the band will become visible as the beads collect there. If there is no antigen on the beads, they'll just continue on through the strip and you won't get the visible strip. Because false negatives can occur with this test, this should always be followed up with a culture. When culturing for strep, you want to use sheep blood agar. In this case, sheep blood agar is both enrichment and differential. It's enrichment because many of the streptococcus are fastidious and they need the extra enrichment found in sheep blood agar. But because the pathogenic organism is beta hemolytic, we use it as a differential media so that we can see the hemolysis of the organisms. Throat culture should be incubated at 35 to 37 degrees centigrade for 18 to 24 hours. That's pretty standard. However, The organisms that live in your mouth are microaerophiles. To be more specific, they're capnophiles. They like less oxygen in their atmosphere and a little more carbon dioxide. So we need to give this culture a 5% carbon dioxide atmosphere. In order to do this, we can use something called a candle jar. And a candle jar is pretty much what it sounds like. It's a large jar with a candle in it. You place the plates in the jar. You put the candle in, you light the candle, put the lid on the jar, and let the candle burn out. As the candle burns, it consumes some of the oxygen in the atmosphere, and the burning replaces some of that oxygen with carbon dioxide. You'll get about a 5% carbon dioxide atmosphere before the candle goes out. Then the entire jar can be placed in the incubator to get the temperature right. To obtain your specimen, you'll need a sterile swab and a tongue depressor. In the clinical setting, you usually use a brand called the Culturette. The tongue depressor helps hold down the tongue, and if you have your patient say ah, it sort of flattens the tongue, making it easier to get to the back of the throat. Strep throat usually causes redness in the throat and sometimes even pus pockets. So if you see that, that's what you want to swab, that red area or that pus in the back of the throat. You don't want to touch the roof or the sides of the mouth or the tongue. If you're using a culturette at this point, you would return the swab to the sleeve and crush the ampule of transport media, and the culturette would be sent to the lab for culturing. If you're using a plain swab, as we will be doing in the lab, you'll want to inoculate it directly to media. To inoculate the media, you roll the swab across the sheet blood agar 2 or 3 times. You want to make sure you roll it so that all surfaces of the swab come in contact with the plate. Then with a sterile inoculating loop, you want to streak the inoculum over half the plate. So you go into that area where you roll the swab and spread that out over about half the plate. Then, just like when you did the isolation technique of the streak plate, you'll sterilize your loop, streak once into that original streaking, and then streak a quarter of the plate. Sterilize your loop, streak once into that second streaking, and streak the last quarter of the plate. You will place the plate in a candle jar, light the candle and allow it to burn out, and then place the jar in the incubator for the appropriate time. When you examine the plate for growth, most of your normal flora will be alpha hemolytic, so you should see lots of alpha hemolytic colonies. Streptococcus pyogenes, the pathogen that we're looking for, is beta hemolytic. So if you see beta hemolytic colonies, those should be further tested to see if they are Strep pyogenes. Now, 1 or 2 beta hemolytic colonies in a sea of alpha hemolytic colonies would not be cause for concern. But if the culture was primarily beta hemolytic, that would indicate strep throat.